Thank you, Rhonda. Can you hear me okay?
Yes, we can. Thank you.
Perfect. Thank you. Hello, everyone. Thank you for joining our webinar. My name is Helly Pimentel, and I'm a product manager here at ACD. My colleague Miguel Mata, an R&D scientist, will be co-hosting with me. In today's webinar, we're delighted to introduce you to ACD's newest product offering called the miRNAscope assay, specifically designed for the detection of small non-coding RNAs, such as microRNAs, and short therapeutic oligos, such as small interfering RNAs and antisense oligos. On the agenda for today, we'll first introduce you to ACD's product portfolio for the use of a wide range of applications. Then we'll switch gear and introduce you to the newest miRNAscope assay to detect smaller RNAs. After that, my colleague Miguel will present on the common applications that necessitate the use of the miRNAscope assay, along with some beautiful data to share with you. Additionally, we'll look at the assay controls and provide outstanding scoring recommendations. Towards the end, we'll talk about the product offerings as well as in-house assay services that are available for your research. Finally, we'll summarize our presentation and address some of your questions live. To begin, ACD's core technology is the RNAscope technology. RNAscope is an ideal spatial analysis solution to interrogate complex tissues. It is a highly specific and sensitive method to detect RNA biomarkers in cells and tissues with morphological context at the single-cell resolution. The RNAscope technology consists of three parts: a unique target probe that ACD designs against your sequence of interest, a signal amplification system that generates a high signal-to-noise ratio, and lastly, visualization of single RNA molecules as dots for quantitative analysis. The assay allows for the spatial mapping of mRNAs, long non-coding RNAs, splice variants, highly homologous sequences, and permutations in cells and intact tissues, all of which can be visualized with either fluorescent or chromogenic labels. In addition, the assay can be performed on a wide variety of sample types, including FFP tissues, fresh frozen or fixed frozen tissues, and culture cells. In this slide, we're looking at the three unique assays that ACD offers to detect any RNA with spatial mapping at single-cell resolution. First, on the left, RNAscope is designed to detect any mRNAs or non-coding RNAs that are greater than 300 nucleotides in length. RNAscope probes are designed in about 20 ZZ probe pairs, and the assays are available in either chromogenic or fluorescent detection modalities for up to 12 targets simultaneously in a single slide. On the other hand, the BaseScope platform listed here in the middle uses probes that are designed in 1 to 3 ZZ probe pairs to specifically support the short target detection of splice variants, highly homologous sequences, point mutations, or short targets that are 50 to 300 nucleotides. The BaseScope assay can detect up to 2 targets with the chromogenic assays. Last but not least, ACD offers the newest miRNAscope technology to detect even the smallest RNAs and oligos, such as microRNAs, antisense oligos, small interfering RNAs, or other smaller sequences in the range of 17 to 50 nucleotides. This new assay is currently available as a chromogenic singleplex assay, which we'll get into more details in later slides. Next, focusing on the core RNAscope technology workflow. The RNAscope assay workflow is similar to immunohistochemistry, so researchers who are familiar with tissue-based protein assays find it quite intuitive to adopt this workflow. First, we start with properly permeabilized cells or tissues with ACD's pretreatment reagents. Then we hybridize the ACD probe to target RNA of interest. Next, with addition of the proprietary amplification system in the form of detection reagents, which allows the visualization of a single RNA molecule in the form of dots and enables us to quantify the level of gene expression indirectly. Our products cater to each of these steps, providing ready-to-use reagents in most cases. To design a specific probe for your target of interest, ACD requires you to provide us with either the accession number information or the exact sequences of the target. You could even indicate your region of interest for targeting with start and/or stop positions. Our bioinformatics scientists channel this information through the proprietary ACD alignment algorithm to select optimal sequences to ensure target specificity and uniformity of oligo hybridization in the assay. Our algorithm evaluates the sequence composition of each oligo in the pool to ensure no cross-hybridization with non-target RNA under stringent RNA conditions. The highlight of the RNAscope technology is our probe design strategy. We can design probes for virtually any species and have done so in over 200 species, covering most applications highlighted on our website. The majority of our probes are designed in human and mouse. On our website, you'll be able to find the applications area to demonstrate how these probes are used in the most common applications, including infectious diseases, cancer, neuroscience, and gene therapy. We've also added support for signaling pathways such as Wnt or JAK-STAT and indicated the probes we have available within that pathway. Now this slide summarizes all the assays that are currently available from ACD. We have chromogenic singleplex brown or red assays and duplex assays on both the manual and automated platforms. The multiplex fluorescent assays allows for the detection of up to 4 targets simultaneously in a single tissue section. The major difference between the two fluorescent assays is that the V2 assay uses fluorophores from Akoya Biosciences for signal visualization and provides a high signal-to-noise ratio, whereas the fluorescent multiplex assay is an all-in-one kit. The V2 fluorescent assay is also available on the Leica automation platform. For higher plexing, we have the RNAscope high-plex assay, which enables the detection of up to 12 targets simultaneously in a single section. Currently, the high-plex assay is available in the manual format only. If your target of interest is shorter than 300 nucleotides and studying splice variants or point mutations or highly homologous sequences, then the BaseScope assays will be the right choice for you. Currently, the BaseScope singleplex assay is available on all platforms, while the duplex assay is available on the manual platform. Finally, expanding our product portfolio, not only do we have assays to detect mRNAs, long non-coding RNAs, short target splice variants, and point mutations, but we now have the miRNAscope assay for the detection of microRNAs, small interfering RNAs, antisense oligos, or other smaller RNAs as low as 17 nucleotides in length. As of today, we have over 26,000 unique target probes in our catalog. Custom probes can also be designed against any targets from any species in as little as 2 weeks. In the next few slides, we'll begin our focus on the newest miRNAscope assay. Overall, the miRNAscope assay is a robust in situ hybridization assay, enabling highly sensitive and specific detection of small non-coding RNAs such as microRNAs and short oligos such as ASOs and siRNAs in tissues with spatial and morphological context at single-cell resolution. In addition to these key applications that I just mentioned, the assay will also enable the detection of any smaller RNAs that are 17 to 50 nucleotides. Some popular targets include tRNAs and pVRNAs. Key research areas for these top targets include cancer research, infectious diseases, neuroscience, and therapeutic target validation. Additionally, this new assay leverages ACD's core amplification system and probe design capabilities to enable the detection of RNA molecules at single-cell resolution while improving high signal-to-noise detection of smaller RNAs with ease of data interpretation. To enable further insights into your research, the miRNAscope assay is also compatible with immunohistochemistry or immunofluorescence assays, allowing the detection of these small RNAs and protein simultaneously on the same tissue slide. Next, what are the specifics of the miRNAscope probe design that makes the assay highly sensitive and specific? So the miRNAscope probe design is a brand new design strategy, but leveraging our core RNAscope technology, that the design algorithm screens through the whole transcriptome to avoid any cross-hybridizations. Due to the extremely short sequences of these targets, sometimes cross-hybridization is unavoidable, so in the cases of potential cross-hybridization, this information will be fully disclosed to our users for evaluation. However, I would like to mention that the majority of the cross-detection targets are endogenous transcripts, so we highly recommend our users to always include a negative control as the baseline to interpret their data. For microRNAs with cross-detection, we recommend the use of a non-negative control sample for the microRNA target. On the other hand, if your targets are ASOs or siRNAs, we always recommend your studies to include a mock or untreated sample for your data interpretation. In addition to the new probe design strategy, we also improved the probe hybridization buffer to deliver optimal assay performance. Together with these conditions, the miRNAscope assay offers superior sensitivity and specificity without the need to further optimize the target probe hybridization conditions. For the miRNAscope assay, we also offer a universal scramble sequence probe as the negative control to evaluate specificity of the assay. Additionally, you could also request a specific scramble sequence to be designed against your small RNAs. As I just mentioned, one of the key features of the miRNAscope assay is the probe design. The oligonucleotide target-specific probes are designed using an informatics algorithm that selects sequences to specifically bind to the target sequence and not cross-hybridize with any other sequences. The bottom of the probe design complements and hybridizes to the target transcript, whereas the top of the probe is the base for the amplification structure, upon which a preamplifier can bind and the amplification tree can then be built. Once the preamplifiers are hybridized to the target probe, multiple amplifiers are then added to the preamplifiers. Each amplifier can then further bind multiple label probes, sequentially hybridizing to assemble a branching complex at each probe binding site. Label probes contain a chromogenic enzyme that generates a visible signal after chromogenic reaction, in this case with fast red, that is detectable under a standard bright field microscope. This entire amplification system allows for highly sensitive and specific visualization of target RNAs as punctate dots. Next, we'll focus on the overview of the miRNAscope assay workflow. The new miRNAscope assay workflow is very similar to the RNAscope singleplex workflow for those of you who are familiar with our technology. First, the cells or tissues that are bound to a slide is permeabilized using ACD's ready-to-use pretreatment reagents. The new assay supports a variety of sample types, including FFPE, fresh and fixed frozen, and culture cells. Then the small RNA target probes are hybridized to the target RNA. Currently, the miRNAscope assay allows for a single RNA target detection. The probe hybridization step is universal across all targets, meaning further probe optimization is not required. After probe hybridization, the target probes will be hybridized with ACD's modified branch signal amplification tree, consisting of amplifiers and label probes to provide high sensitivity and specificity. After the colors have been developed, the small RNA molecule signals can be seen as red chromogen deposits using bright field microscopy. I would also like to mention that the fast red chromogen can also be visualized under fluorescent microscopes, similar to the ATO 550 dye. The slides can then also be scanned for further qualitative or quantitative analysis. The entire singleplex assay workflow can be done either manually or fully automated using the Leica systems. Switching gear, as I mentioned in a previous slide, that the miRNAscope assay is compatible with multiple tissue types. In this slide, we're demonstrating robust assay performance in various species of formalin-fixed paraffin-embedded tissue samples. For example, demonstrated here, microRNA 124 is detected in mouse, rat, and human brain tissues with abundant signals, To demonstrate specificity, a known miRNA 124 negative tissue, such as the mouse liver, is also stained with the miRNA 124 target probe. As you can see here, the negative tissue is very clean with no detection of the miRNA 124 target probe. Additionally, the miRNAscope assay demonstrates similar detection robustness with fresh frozen tissue samples. Demonstrated here in the left column, miRNA 124 is stained in the fresh frozen mouse brain tissue with abundant signals detected similar to what we just saw with the FFPE tissues. Again, the scramble probe is used as the negative control to demonstrate assay specificity. On the right column is another example of fresh frozen tissue, but with miRNA 122 stained in the mouse liver, showing robust detection of the target and no background with the scramble negative control. On the other hand, if you're working with culture adherent cells, the miRNAscope assay also performs extremely well, as demonstrated in this slide, with miRNA 205 stained in A431 skin cells. The assay picked up the abundant miRNA 205 signals in the skin cells, and again, the scramble probe is negative. Overall, the miRNAscope assay is a highly robust assay that can work with any of your sample types. So why choose ACD's miRNAscope assays? The miRNAscope assay offers many benefits for your research. First of all, the assay enables highly specific and sensitive detection of your small RNA targets at single-cell resolution, while preserving spatial and morphological context. Additionally, the assay is compatible with multiple sample types, including FFPE tissues, fixed and fresh frozen tissues, and culture cells. Another benefit of the miRNAscope assay is that it does not require an RNase-free environment like other typical assays do. Furthermore, the assay reagents are offered in ready-to-use formats for ease of use. On top of that, ACD also offers a one-on-one scientific consultation with our expert technical support scientists to ensure you have a successful run of the assay. In addition, the miRNAscope assay is compatible with IHC staining, enabling co-detection of RNA and protein in a single tissue slide. Last but not least, the miRNAscope assay uses universal assay conditions where minimum optimizations are required for a successful detection of your target. Prior to commercialization of the product, we have many successful customer stories using the miRNAscope assay through our in-house pharma assay services team. For example, one of the customers had used the miRNAscope assay to detect their siRNA and provided us with an extremely valuable feedback, saying that the assay is incredibly specific and that the results are very easy to understand. The ACD Pharma Assay Services team had generated impeccable tissue staining for their siRNA therapeutic research and development. Another successful customer who had used our miRNAscope assay to detect their ASOs says that they use ACD's assays to detect both the RNA and short oligos in serial tissue sections, which generated signals that are both evident and specific. And with that, I'll hand it over to Miguel to provide you with detailed presentation on how the miRNAscope assay could help with your research.
Hello, can you hear me okay?
Yes.
Great. Okay, well, thank you so much, Helly, for that great introduction on miRNAscope. This product was just launched last week, and so we're very thrilled to be able to present to you some of the exciting results that we've been able to obtain in-house, as well as from various assay services projects that we have completed. So with that, we'll start with this first slide. So what are the key applications that miRNAscope enables detection? So if we focus on, as Helly mentioned, miRNAscope enables detection at a single cell resolution of small RNAs. This RNAs can be synthetic or endogenous, so between 17 to 50 nucleotides. In this category, there's a number of synthetic oligos that have gained notoriety in the past few years for their potential as therapeutic agents. And this include antisense oligonucleotides, commonly referred to as ASOs, small interfering RNAs, siRNAs, and also short hairpin RNAs. These three synthetically synthesized RNAs, as I mentioned, have the ability to have shown to have activity in the clinic by regulating transcripts that are in cases upregulated in certain pathological conditions. Also, another particular application for miRNAscope assay is the detection of endogenous microRNAs. So microRNAs are endogenous small RNAs between 18 to 24 nucleotides in length, and they have been shown to regulate various signaling pathways all the way from development all the way to adulthood. And also in the past few years, there's been implications of this microRNAs in cancer and other diseases, and there have been there's potential avenues for using them as biomarkers for different disease conditions. Also, shRNAs, piRNAs. So these are small RNAs, but they're between 26 to 34 nucleotides in length. And one of the main functions of this small RNAs is that they silence transposable elements within the germline. So you can think of them as gatekeepers of the germline and fertility as well. So this particular RNAs have also been observed in a number of diseases such as cancer, and their function and mechanism is still widely underappreciated. In terms of the applications, the key research areas for the miRNAscope assay, the first one includes basic science in fields of neuroscience, stem cell, and developmental biology, and we'll be talking about a few examples from the miRNAs perspective. A second is infectious diseases in terms of inflammation and immune cell regulation, and this has been particularly important in terms of viral infections. And we'll discuss a few microRNAs that are involved in immune cell regulation. Next, biopharma therapeutics. As I mentioned, siRNAs and ASOs have gained a lot of attention for their potential to be delivered in a very tissue-specific manner and have the potential to downregulate key players within signaling networks that are amplified or deregulated in various conditions. And for this, miRNAscope assay has allowed siRNA and ASO studies into delivery into the chemistry, into the biodistribution, into the toxicology, and also the stability of this small RNAs. Finally, cancer and clinical research. As I mentioned, all of these small RNAs have the potential to be deregulated or impact a particular condition, such as they can be used in tumor biology, immuno-oncology, as well as pathology. So let's move on to the miRNAs applications. So there are a few key research applications where miRNAscope could play a significant role in detecting microRNAs. The first is oncology, and in oncology, in particular in oncology, microRNAs in the past have been shown to alter the transcriptional landscape within single cells. And in this way, it induces and may induce malignant transformation of those cells. So there's some interest in targeting particular microRNAs because of their characteristics of promoting malignant transformation, and they're commonly referred as oncomers. On the other hand, polar opposite, there are other microRNA targets that have tumor suppressor characteristics, and there's a number of companies that are interested in delivering either some sort of molecules to target this microRNAs in this particular oncology field. Next, in neuroscience. So neuroscience, there's numerous microRNAs that have been previously involved in pathogenesis in various neurological disorders. And today we'll show you some of the microRNAs that we've been able to identify in some of our model systems to show you the specificity and also the exquisite sensitivity of the assay. Finally, the last application that we'll be discussing is immunology. As I mentioned, microRNAs have the potential to regulate immune cell activation, and this is in particular important in the case of viral infections. As infections proceed, the immune system tries to react, and microRNAs have been shown to play a role in this way. So next, I'll talk about some specific samples of microRNAs in the oncology research area. So here, what we're showing are various microRNA targets, which were, of course, detected using our miRNAscope assay. And these microRNAs have already been reported to play a role in these two types of cancer. First is cervical cancer in the top row, and bottom we have head and neck cancer. So microRNAs in these two different cancers are known to deregulate signaling networks that promote malignant transformation. And such an example of this is miR-205-5P, which is on your far left. This particular microRNA is known to promote tumor invasion, migration, angiogenesis, and one of its key targets is AKT activation. So by activating AKT, the proliferation of cells is activated, and initiation and progression of tumor starts. Next is miR-21-5P, and this is one of the earliest identified cancer-promoting microRNAs, or as they commonly refer to them as oncomiRs. And the way miR-21-5P acts is by activating the PI3-AKT pathway, again, activating cell proliferation, and also by enhancing oncogenic KRAS activation. Two microRNA targets that have been widely studied are miR-155-5P and miR-223-5P in the context of the immune response to various conditions, including viral infections as well as cancer. In the case of miR-155, this is an immune-enriched microRNA, and it's particularly expressed in cells of the lymphoid origin, for example, T cells and B cells. miR-223-5P, on the other hand, is also an enriched microRNA in the immune system, but it is commonly observed in cells of the myeloid origin. So cells such as macrophages and neutrophils tend to express miR-223-5P. And what you can see is that these two microRNAs are observed in these two cancers, suggesting infiltration of these key cell types, macrophages potentially, and T cells. And they're observed at differential expression levels. As a control, we have scramble, as Helly also had shown previously. And this scramble provides us with a way to show that the staining that we're seeing is actually a true signal and not an artifact. We're seeing very clean background in terms of the scramble signal. Next, another key application is miRNAscope-ish assay in combination with IHC detection. So as mentioned previously, this miRNAscope assay is fully compatible with immunohistochemistry. And this is in an effort to allow researchers to identify specific cell types in tissues of interest, or even to measure the levels of a particular microRNA target. And in this way, we have enabled miRNAscope initial detection on both manual as well as on the Leica platform. Now, this capability has been optimized. And if you already have pre-established RNAscope IHC parameters, you can transfer those same parameters and apply them to miRNAscope. So what we're seeing here in this particular slide is a couple of things. The first in red is miR-223-3P, which I mentioned is a myeloid-specific microRNA. And the other in IHC, looking at protein marker, is CD3, which is a marker of T cells. So one thing that we can see is that both the CD3 and the miR-223 signal are mutually exclusive, as is expected, given that T cells come from a lymphoid origin and miR-223 is expressed in a myeloid-specific manner. Our next example is moving into neuroscience research area. So Helly touched on miR-124 briefly in her previous slides. But this is a very nice image showing the miRNAscope ability to detect miR124 in a very nice section of the mouse brain. And we're looking at miR124, and you can observe that it's one of the most highly enriched microRNAs in the brain. And miR124 actually plays a crucial role in supporting neuronal differentiation. And one of the ways that it does this is by targeting PTBP mRNAs. And in this way, it allows for pre-mRNA splicing to continue. And you can see several regions in which there's particular enrichment compared to other regions. And these are particularly important to study and understand how miR124 is playing a role in those particular areas. Next, we're seeing miR124 across a variety of tissues. Again, showing how miRNAscope assay has the potential to be used across species and how robust this particular assay is. So first, we have on the left is a mouse brain, which we've shown a few images. And you can see here very nice expression in the hippocampal region. Next is the rat brain. In the rat brain, we see strong enriched signal in the cerebellum. And this is a particular human brain section that we're also seeing very nice staining of miR124 3P. Now, as Helly mentioned, we use a mouse liver sample, which is our negative control tissue. And this shows the specificity of the probe. And in this way, we have been able to validate multiple microRNAs by looking at positive and negative tissues to be able to distinguish the true signal from potential background. Next, this is also in line with neuroscience. We're looking now at other microRNAs that have been studied previously, and there's substantial literature research on these microRNAs in terms of their role in the development of the mouse brain. So in the first column, we have miR124, which we have discussed already, but on the bottom panel, bottom row, we can actually see a zoomed in image of the signal that we're seeing, and you can see it's a very enriched microRNA. The next microRNA we have is LET7B5P, and this is part of the very famous LET7 family of ultra-conserved microRNAs. And LET7B has been shown to play numerous roles across all tissues. And in this particular case, LET7B is involved in central nervous system pathologies, as well as tumor growth and even immune responses. So what we're seeing here is in the cerebellum, we're seeing very strong staining of LET7B5P. Next, we have miR138-3P. And this is one of our favorite microRNAs because it shows you the spatial resolution of this particular assay. It provides you with that morphological context that you absolutely need in order to understand the function of this particular microRNA. So miR138 is particularly expressed very exquisitely in Purkinje cells and in the cerebellum of the brain. And this microRNA actually plays a very important role in cellular differentiation and maintenance of this particular cell type. Next, we have a cortical region expressed microRNA, miR128, that we're seeing here. In this particular microRNA, there is ample evidence showing that it plays a significant role in neurogenesis and synaptogenesis from very early stages in development, at least from research done in the mouse, all the way to neural migration and outgrowth. And this is a microRNA that we can visually observe in this particular sections. Again, to show the level, to show that this is in fact true signal, we're using our scramble probe showing no signal in the same areas where we're looking in the mouse brain. Now we're moving into a different key application of miRNAscope, which is developmental biology. So this particular image is quite striking, and it's perhaps one of the favorites because of the resolution that you get to observe from detection of miR124. So in here we have a mouse embryo, age E15.5, and this is a sagittal section. So on the top, we have the embryo's head, and towards the middle and the bottom, that's the embryo's body. Now, this particular slide is showing very nice detection of miR-124, specifically in the brain and not in other regions, or in the central nervous system and not other regions of the mouse embryo. And to show the specificity of this assay, we have again used our scramble probe to demonstrate the robustness of miRNAscope. And we can see very strikingly how miRNAscope delivers that spatial resolution that is required in order to fully understand the function of these microRNAs. Another microRNA in developmental biology that has been widely studied is miR-145-5P. And again, this is another embryonic sagittal section of an embryo at E15.5. And what we can see here are three different areas within the embryo. The first area is the digits, second column is the somites, and then the third is the ear. This particular microRNA is implicated in cartilage development and is specifically expressed in cartilage tissue. There's also very important evidence showing that one of the targets of miR-145P, which is fucosal transferase, or short for FUT1, it's implicated in osteoarthritis. And this particular disease is characterized by cartilage degradation and chondrocyte injury. What we observe here is quite a striking differential expression of miR-145P across the different tissues, which includes, again, digits, somites, and ear. And to demonstrate that this signal is specific, we're showing here a scramble image showing no signal from the same areas in which miR-145P was detected. Next, we have microRNA detection in the mouse eye, specifically in the retina. So this image is showing miR-182-5P. And miR-182-5P is specifically expressed in the outer nuclear layer of the mouse retina, and which is the red chromogen signal that we're seeing at the top of the image. Now, what this image is showing is very specific detection in this layer, because subsequent layers, which include the inner layer and also the other layers of the retina, show very little to no detection of miR-182-5P. So as you can see, there's microRNA 182 can be highly detected in this particular tissue. And miR-182 has multiple roles within the retina, including cone receptor and visual function, and also deletion of this particular microRNA in mouse models results in retinal dysfunction dysfunctional in mice, showing that this particular microRNA is very important for the development of the cones and the rods within this outer nuclear layer. Next, we have a microRNA that has been shown to be highly enriched, and it's also very specific to vascular smooth muscle cells. And I'm talking about miR-145-5P. And in this case, miR-145-5P has been shown to regulate these types of cells, and specifically regulating the proliferation and migration. And it does this by targeting two very key signaling pathways, which include SMAD4 as well as the TGF-beta signaling pathway. And because of its role in TGF-beta, there's also several publications showing miR-145 and its role in cancer progression. Now, on this slide, we actually have captured multiple mouse tissues, including the lung, the intestine, and the mouse kidney, to demonstrate the signal that you can obtain using miRNAscope assay. What we can see is very specific detection in this particular cell type, the vascular smooth muscle cells, whereas other tissues are negative for miR-145-5P. Next, another application that we'll be discussing is siRNA detection, or small interfering RNAs. And this particular small RNAs have shown very significant potential in 3 key areas, which include oncology, neuroscience, and immunology. In terms of oncology, this particular siRNAs have been shown to be a great source as a great therapeutic agent, targeting key oncogenes, and in this way downregulating key signaling networks in different cancer or tumor growth. In terms of neuroscience, there's research and also a lot of siRNAs that are moving into the clinic, showing that siRNAs can be used in clinical treatment of acute brain injuries, as well as neurodegenerative disorders. And finally, in terms of immunology, siRNAs can regulate or induce some sort of immune response to a particular immune disorder, or even target particular genes that are deregulated in autoimmune disorders, and in this way mitigating the disease progression. So this is an example of a recent project that we completed through our assay services at ACD. And this is a very striking slide because it shows the specificity of the assay. So in this particular project, 3 cohorts of animals were delivered either saline, or these are control negative control animals, which is on the left. A different cohort was delivered with the siRNA, which we're calling siRNA1 for purposes of this presentation. And a different cohort was delivered a independent siRNA molecule called siRNA number 2. And using the siRNA number 1 probe, we can actually see specific detection of siRNA1 in the tissue that we should be observing detection, which is the tissue in the middle. Whereas the 2 tissues on flanking siRNA number 1 are negative. So this sort of data, this type of actionable data can now be used and then address other key things related to siRNA. For example, is there any particular modification that would be beneficial? So you can examine biodistribution of the siRNA as a function of different modifications. You can also examine the stability of siRNAs after a dose, increasing dosage or titration of a particular siRNA. And also you can study the impact of this particular siRNA as a function of the target that you were focusing on. So there are multiple applications for miRNAscope in terms of siRNA detection. Next is ASOs, and this is in the small oligonucleotides, antisense oligos. And there are 3 key research areas, which include oncology, neuroscience, as well as immunology. In terms of oncology, ASOs have been, as I mentioned, they're synthetically designed, and they target and they have the ability to block oncogene expression. And in this way, you can deregulate or slow down the growth of a particular cancer type. In neuroscience, ASOs have emerged as an important therapeutic strategy, and ASO therapies are now being used to target genes that were a few years ago thought of as undruggable. So many ASOs are actually moving into the clinical trial pipeline, and there's even some ASOs that have already gained FDA approval for treatment of a number of neuromuscular disorders. And finally, we have synthetic oligonucleotides that can target immune regulators, and in a number of diseases, which include autoimmune disorders. Again, this is a project that we recently completed through the assay services at ACD, and it's showing an example of miRNAscope assay specifically detecting the antisense oligonucleotides. In this case, what we have here are 2 types of tissue, mouse cardiac muscle and mouse quad muscle. Now, 2 different cohorts were delivered either a vehicle or were treated with the ASO here that we're trying to detect. As you can see, the ASO probe specifically detects the ASO treated animal, as can be shown here by the red chromogen signal, whereas no signal can be observed in the vehicle treated sample. Again, using this information, one can then ask more questions. So this is actionable data that you can act on, formulate new hypotheses, and try to understand how you can improve antisense oligo delivery by adding modifications, conjugating the ASO to be delivered specifically to a particular tissue of interest. You can also study biodistribution. Is it expressed in a particular tissue, or is it more systemically delivered into the different tissues within the animal? And the other questions that can emerge from here. Finally, we arrived at our last application, which is detection of piRNAs. There are two key applications. One includes epigenetics, and the other one includes oncology. Now, piRNAs are actually considered the largest class of small non-coding RNAs, and they're expressed in animal cells. And in contrast to microRNAs, which are between 18 to 24 nucleotides, piRNAs are slightly bigger, between 24 to 36 nucleotides in length. Now, piRNAs, in terms of epigenetics, are associated with their effector proteins, which are Pw proteins. And in this way, they're guarding the germline of animals. And they do so by maintaining the integrity and also fertility from mobile DNA elements, which are also known as transposons. In terms of oncology, there's actually a number of piRNAs together with their effector proteins that have been shown to be abnormally expressed in numerous cancer types. However, their function and the mechanism by which they function within the cancer is it's still largely unknown. And we believe that this will be a key application to look into piRNA function. Here, we're showing the detection of three independent piRNA molecules. We have piRNA 1, 3, and 13, 11, 21. And we're looking at the mouse testes. And these are already published piRNAs that have been validated, and they interact with Pw proteins. So we know that these are piRNAs, in fact. And these are germline specific, so they're not expressed in any other tissue besides the germline. And the mouse testes is our model to study this. Now, piRNA detection in this case appears as a very strong paranuclear pontic dot. And as you can see from this image, is specifically detected in the spermatogonia, which is this undifferentiated male germ cells that eventually give rise to the mature spermatozoa. Now, these staining patterns are also have also been observed in multiple literature reports. And as a negative control, we're showing here a scramble probe, which shows no staining in the same area in the mouse testes. And also to demonstrate further the specificity of these probes and overall miRNAscope assay, we have here a mouse liver in which we stained it with the piRNA 13, 11, 21. As I mentioned, these are germline specific piRNAs, and we actually see no signal from mouse liver, which is a somatic tissue. Again, demonstrating the assay's specificity and also its sensitivity, giving single cell level resolution. And with that, I'll pass it on again to my colleague, Helly.
Great, thank you, Miguel. Can you hear me?
Yes, we can.
Perfect, thank you, Rhonda. So thank you, Miguel, for the excellent application demonstrations. And in the next few slides, I'll talk about our recommended assay controls and scoring guidelines for the miRNAscope assay. The miRNAscope assay to detect small RNAs will offer universal positive and negative control probes that will work for any species or samples. For the positive control, we recommend the small nuclear U6 probe, which is known to be expressed in most tissue samples. For example, on the top row here shows a strong nuclear U6 signal observed in a variety of tissues, including human and mouse tissues, and HeLa and 3T3 cell pellets. On the other hand, as you've seen from Miguel's data, a universal scramble probe is recommended as the negative control probe to demonstrate assay specificity and to be used for data interpretation against your target. Next, we'll focus on the miRNAscope scoring recommendations. The miRNAscope assay data could be analyzed with either semi-quantification or single-cell analysis with a software analysis tool such as Helly or other open-source software analysis tools. The semi-quantification scoring criteria shown here is specific for the miRNAscope assay, which provides guidelines for your target expression levels as counted by dots per cell. If your target is highly expressed, such as with 20 dots or more per cell, then we recommend a score of 3. Whereas your target is moderately expressed with 11 to 20 dots per cell, a score of 2 is assigned. For lower target signal detected, such as 2 to 10 dots per cell, a score of 1 is assigned. Lastly, a score of 0 is assigned if there are no staining or less than 1 dot per cell observed. A note that we want to mention, though, is that due to the relatively small size of the microRNA or ASO siRNA targets, the puntate dots seen under the microscope may represent multiple copies. As a result, we recommend our users to interpret the data by relative quantification, meaning that they should always use the negative control as the baseline for measuring the change in their target probe expression. Next, I'll go over the miRNAscope products that are available. As mentioned previously, the miRNAscope assay is offered in both the manual and LIGO formats. Each assay with universal control probes that are species independent, but are platform specific, so be sure to request the correct catalog probe for the assay platform that you prefer. For the manual assay, we have the HD reagent kit that includes all the pre-treatment reagents, along with the detection reagents needed for the assay. Additionally, we also offer 2 introduction packs for the most popular species samples, such as human and mouse, for your convenience, where the packs include the pre-treatment reagents, detection reagents, control cell pellets, mounting media, and a hydrophobic barrier pen that are necessary to complete the entire assay. On the other hand, for LIGO users, we have the LS reagent kit, where it includes the pre-treatment reagents, wash buffer, and detection reagents. The miRNAscope target probes are designed in the S1 channels that are specific for this assay and cannot be interchangeably used with the other assays such as the RNAscope or BaseScope assay. We're very delighted to also share with you that we have a rich list of miRNAscope probes that are pre-designed as catalog probes, so you could request them immediately. So feel free to reach out to your local sales representative or to our support team for the list of probes available. For custom design requests, we request you to submit your target through the ACD's new probe request form through our acdbio.com that is listed on this slide. In addition to products, we also offer in-house assay services for those of you who would like to send in your samples. So we understand the challenges that many of you may be facing during the pandemic. However, research must go on. That's why our services team are here to assist you with moving your research forward. Our pharma assay services are fully operational and could help you advance your research in the detection of microRNAs, antisense oligos, and siRNAs if you would like to send in your samples and prefer the assays to be validated by ACD miRNAscope assay experts. Our services teams ensure that all due care is taken with your samples and that the assay development and analysis are conducted with highest efficiency. Our pharma assay services have recently received awards from the Pharma Tech Outlook for being one of the top 10 CROs of 2019 and 2020 consecutively. In addition to pharma assay services, we also offer academic assay services for those of you who are working in the institutions. The benefits of having ACD validate your target of interest in your samples includes saving resources with your pilot study before setting up for a larger scale in your own labs, and receiving your results in a quick turnaround for your grant application and or paper submission. So for further information, please contact our support team with the email address listed here. So with that, we'll summarize today's presentation. So our new miRNAscope assay is a robust RNA in situ hybridization assay, enabling the detection of small RNAs in tissues with spatial and morphological context at single-cell resolution. Key applications of our new assay include ASOs, microRNAs, siRNAs, or other short RNA sequences that are 17 to 50 nucleotides. The new assay is a singleplex chromogenic fast red assay as it's compatible with the manual platform or the fully automated Leica platform. The miRNAscope assay is also compatible with IHC or IF staining for the visualization of RNA and protein simultaneously on your tissue. Again, in-house assay services are also available for this assay if you like our expert scientists to perform the assay for you, especially during the current pandemic. So with that, we would like to thank everyone for attending today's webinar. For further details of ACD's newest miRNAscope assay, please visit our website or contact your local representative to learn more. You could also contact our technical support team for any questions that you may have regarding the new assay. The support contact information is listed here. So next, Miguel and I will be addressing some questions from the audience. So Rhonda, would you please read out some of the questions that we received?
Hi, Helly. Thank you so much, both Miguel and Helly. What excellent presentations you presented today. That was really, really good. We have some fantastic questions coming in. Our support team and panelists are working hard to get them all addressed for you. I do have a couple of questions here that I can address with you both. But in the meantime, like I say, just stand by. We are getting to all of your questions, and if we don't get to them today, we will make sure that you get your responses via email after the event. First of all, for Helly, do you have a list of target probes available for purchase? And can you, if so, can you also design probes for custom sequences?
Thank you, Rhonda. Yes, we do have a list of miRNAscope probes that have been pre-designed, and also most of them have been validated in-house. They could be purchased immediately. You could also submit custom sequences for probe design feasibility through our ACD Bio new probe request form.
Sounds great. A question here. Another one for you, Helly. Is the assay protocol different from the RNAscope assay?
Sure. So the miRNAscope assay protocol is very similar to the RNAscope protocol. Specifically, the manual miRNAscope protocol has an extra fixation step to ensure optimal detection of these smaller RNAs, whereas the Leica protocol is very similar and straightforward, similar to the RNAscope Leica singleplex assay that can be completed with an overnight run.
Okay, thank you. And then this one popped in for Miguel. Is the assay compatible with protein detection, such as immunohistochemistry or immunofluorescence? We actually got that a lot in our Q&A as well, so great question.
Correct. Yeah, this is a great question. The answer is yes. miRNAscope WORKFLOW is fully compatible with IHC or IF, and this is for both the manual and also the Leica offering. As I mentioned previously, if RNAscope IHC conditions have already been predetermined to be optimal for a particular sample type, those same conditions can be transferred and applied using the miRNAscope assay. So there's no additional need for optimization as long as you have predetermined those conditions.
Okay, great, thank you. And then there's another one here for you, Miguel. How do you determine specificity of the miRNAscope target probes?
That is a great question, and also one that we also continue to receive. So the miRNAscope assay, in fact, leverages the core technology behind RNAscope probe design, and also its background suppression system. And in this way, we ensure that there is high specificity. In addition, all of the target probes are actually screened through the whole transcriptome of any particular species of interest. And in this way, to try to avoid any cross-hybridization. We also, as we have discussed throughout the presentation, we encourage customers to include a negative control, either a tissue that is known not to express that particular microRNA of interest, to demonstrate the specificity and also the specificity of the probe that the researcher is trying to study. So this is a way in which the miRNAscope assay delivers the specificity and the sensitivity to the same higher standards that we have for RNAscope and BaseScope.
Great, thank you. And then one more here for Helly. Can the miRNAscope assay detect RNAs that are greater than 50 nucleotides?
Great question. So the miRNAscope assay is specifically designed to detect smaller RNAs that are 17 to 50 nucleotides. So for RNAs that are greater than 50 nucleotides, we will recommend the BaseScope assay, where it supports sequences from 50 to 300 nucleotides. And also for any RNAs that are greater than 300 nucleotides, our RNAscope assay would be the one that supports that.
Okay, great. One question we did get a lot here. Will miRNAscope probes detect mature miRNA and also the pre-miRNA counterpart? We had this question come up quite a few times today. I'm not sure which one. Who can address that?
Yes, I can address that. So the miRNAscope probes detect the mature microRNA sequence. In terms of the premature, and typically the premature form of the microRNA is in lower abundance. So the signal that you would observe, particularly in the cytoplasm, that would be primarily stemming from the mature detection of that microRNA in question. If there is a potential for, as we all know, microRNAs, they're synthesized from the nucleus. If there's potential for nuclei detection, that would indicate the presence of a very, very primordial microRNA form, which is a primary microRNA. But typically what we have observed is that we're detecting the mature sequence, which is cytoplasmic in the majority of the microRNA cases.
Okay, great, thank you. And then one more, there's so many questions actually. Our team is working really hard and we've got some excellent Q&A going on, very active discussion. One thing that just wants another confirmation here is, what are the sample types that can be used for this? Can we, you know, is it specific to fresh frozen, formalin fixed? Like, what's the main sample type this has been optimized to?
Sure, I can answer that. That's a great question. So the miRNAscope assay, similar to our RNAscope and BaseScope assay, is compatible with multiple tissue types. So formalin fixed, paraffin embedded FFP tissues, and also fresh frozen tissues, fixed frozen tissue, as well as culture adherent cells. So all of these sample types are compatible and have been optimized with the miRNAscope assay.
Great, thank you. And we did get a couple of requests from the audience here. Is this presentation available for download? And yes, give us about 24 to 48 hours and we will have an on-demand link on the pre-recorded webinar section of our acdbio.com website. So with that, like I say, we have plenty of questions coming in. Our teams are working hard to answer them. If we don't get to answer them now, we will get back to you via web. And with that, I believe we have finished the live portion of our Q&A today. Thank you both, Helly and Miguel. That was an excellent presentation.
Thank you all.
Thank you, everyone.